Thanks, Vadim, and thanks to the organizers for the kind invitation to present my work here. So I'm going to tell you about the modeling work we've done in epigenetic clocks and how this has led to some new interesting insights. So just to make sure that everyone is on the same page, I'm going to start with a definition of a molecular aging clock. So my definition is that it's a counter that keeps track of age cumulative molecular alterations as measured relative to a suitably defined ground state. And so what that ground state is a very interesting question. And this counter can quantify chronological age or any of various forms of biological age. And this could include all-cause mortality, it could include disease risk, or cellular age, like mitotic age, or, for instance, telomere length. Now, as you can see, I've put one of Dali's famous paintings in the background. Well, my art history teacher taught me, this painting was inspired by Einstein's theory of relativity. And so it's actually quite interesting to contrast physical space-time to biological time, because obviously physical space-time, we've known that it's quantifiable. There have been clocks, astronomical clocks, dating back 10,000 years ago. But the fact that it's relative and malleable was an insight that was only attained about 100 years ago. In the case of biological time, it's actually it's the other way around, right? So we've known for a very long time that it's relative. People of the same chronological age don't die at the same age. And over the last 40 years, we have learned how to modulate it. And it's only fairly recently, with the introduction of aging biomarkers and molecular aging clocks, that we've learned to quantify it, okay? So among all the molecular aging clocks, I think it's fair to say that DNA methylation clocks, they stand out for multiple reasons. So first of all, they're very accurate in terms of chronological age prediction. And one of the reasons why they are so accurate is that you can measure 1% changes in DNA methylation with over 90% sensitivity, right? So I repeat, 1% changes in DNA methylation. You cannot do this with proteomic assays or metabolic assays or transcriptomic assays. There are pan-mammalian clocks, as shown beautifully by Steve Horvath. I think it's also fair to say that they've undergone the most extensive validations in relation to, for instance, all-cause mortality. And if we're talking about solid tissues, they've also been shown to be predictive of cancer risk. And they also predict cellular rejuvenation upon age reversal interventions. However, a drawback has been interpretability. So the way these clocks have been constructed, it seems to be clear that it's sort of a composite of many different underlying processes, and I've listed some of these processes here. And what I would like to do is to focus and tell you a little bit about age-related changes in cell type composition. Okay, so 2 years ago, we published this study in Genome Medicine where we derived a DNA methylation reference panel for 12 immune cell types defined across 600 CpGs. And with this panel, what you can do is then, in conjunction with statistical algorithms, you can estimate the fractions of these 12 immune cell types in any blood sample, okay? As long as you measure the majority of these CpGs. And then you can correlate these immune cell fractions to health outcomes, yeah? Or phenotypes. So for instance, we did this in relation to age. And what you can see here across 20 different cohorts, that the naive CD4 and CD8 T cell fractions decrease with age. So it's a very strong signature. And of course, that is something that we already know about, right? I'm not an immunologist, but Some might call this immunosenescence. Yeah? What is less well known is that these fractions also correlate with health outcomes. So the naive CD4 T cell fraction is protective of all-cause mortality. And so it raises the question, all these associations that we found with the clocks, whether they are driven by changes in cell type composition. Now, we're still addressing that question because to do this properly, you need to characterize all the underlying heterogeneity that exists in blood, which is quite substantial, right? I don't still have an answer to that question, but what we have done is to quantify how much of the changes in cell type composition contributes to the accuracy of an epigenetic clock in relation to chronological age. So we know that there are two processes. There's an extrinsic aging process where you've got changes in cell type composition, and you've got an intrinsic process where you've got changes in methylation in individual cell types. And this could be cell type dependent, right? So the different cell types in a tissue may or may not be aging at the same rate. We don't really know. And so what we wanted to do first was therefore to quantify these two contributions. So how do we do that? So we can estimate the intrinsic fraction by building two clocks. So for one particular cohort, we built two clocks. One which has been so one that has been built not adjusting for cell type heterogeneity, and one that has been adjusted for cell type heterogeneity. And we use this approach, we applied this to whole blood and brain. And what you can see is that about 60% of the accuracy of a clock in blood is driven by intrinsic changes. The rest is then driven by changes in cell type composition, and I've already mentioned the shift of naive to memory T cell fractions. In the case of brain, 90% of it is, or approximately 90% is intrinsic, the extrinsic variation being driven by shifts within the neuronal set. And there's independent validation of this from single nucleus RNA-seq data from Joe Ecker's lab. Okay, so how do we now quantify the intrinsic aging rate of different cell types? So we need to build cell type specific clocks. And ideally you would generate methylation data in purified cell populations, so in sorted cells or at single cell resolution. But as we know, this is not scalable to the hundreds, if not thousands of individuals that we really need to very robustly identify age-associated changes. So currently the best strategy seems to be to take large bulk tissue DNA methylation data sets to estimate the cell type fractions and to then use these fractions to identify the CPGs that change with age in specific cell types. And so over the years, we've published DNA methylation atlas to estimate cell type fractions in various solid tissues, and an algorithm that uses statistical interaction terms to identify cell type specific changes. Okay? So we applied this methodology to liver tissue. So we took a large normal liver tissue data set, and we built a hepatocyte clock, which we then validated in primary hepatocyte cultures. And we also built a liver-specific clock that is, so it's an average over all the cell types in liver. And we compared this to the Horvath clock, and we can see that we get a marginally better performance. We can also show the specificity to liver, so the clock does less well in other tissue types. And we also found the hepatocyte clock to be accelerated in various conditions, so non-alcoholic fatty liver disease, in obese individuals, and also in a genetic condition that increases the risk of cirrhosis. Okay, so we also applied this methodology to brain. We built neuron and glia-specific clocks. We showed the specificity to brain. And we found strong acceleration of the glia clock in the temporal lobe of Alzheimer's disease patients. So there's been a lot of debate about the age accelerations in neurodegeneration, some studies reporting accelerations, and then other studies saying that this was just driven by changes in cell type composition. So having clocks that are more cell type specific now does seem to suggest that we are finding acceleration. And what was really interesting was that when we looked at the overlap of these CPGs, clock CPGs with other clock CPGs, we found a small, but actually it was significantly enriched for CPGs of the dam age Corso clock from Vadim Gladishev's lab. And what was particularly striking, at least to me, was the fact that the few genes that were mapped by these CPGs, they've been implicated in Alzheimer's disease. Okay, so if you're working on neurodegeneration, I'm sure these genes, you know about them. Okay, so let me now tell you a little bit about another aspect that we've been investigating, and that relates to how stochastic the age-associated DNA methylation ages are. So we've got a collaborated Fudan University from which we got a whole genome bisulfite sequencing data set in over 1,000 Han Chinese. And the great thing about bisulfite sequencing, whole genome bisulfite sequencing, is that we get these single reads. And so we can ask the question whether if a particular site changes with age, how likely is it that the neighboring sites are also changing with age in the same cell, because it's the same read, okay? The problem is that it's whole blood, so we don't know which cell type the read is coming from. So what we did was to restrict to CPGs that are stably methylated or unmethylated across all the immune cell types. Of course, we allow for variation with age, but that's fairly small. And to cut the long story short, what we found was that the patterns of age-associated change are non-stochastic up to a scale of around 50 to 80 base pairs. And beyond that, it seems to be quite stochastic. Yeah? That is not to say that you don't have non-stochastic changes globally. Because remember, this is just a local analysis. We're just looking locally around specific HDMCs. So to investigate this question of stochasticity in the context of epigenetic clocks, we took the following approach. So these three clocks are known to vary in terms of their predictive ability for chronological age, or conversely biological age. And so what we then did was to build a stochastic analog of these clocks. And this is done through a simulation process I will describe later. And so these 6 clocks are then applied to whole blood cohorts as well as sorted DNA methylation data sets. And then we compare the predictive ability of the 2 clocks. And then we build this ratio, this RR2 index, which basically quantifies how much of a clock's predictive ability is described by a purely stochastic process. Okay, so the way we do the simulation, you need to start at the single cell level, obviously. I'm not going to bore you with the mathematical details. I mean, this is just a simple 2-state Markov chain model that can recapitulate any sort of dynamics. The key thing to note about this slide is that the effect sizes of HDMCs, as assessed in a purified cell population, a fairly large data set, these are very small. So over 50% of the HDMCs, yeah, display less than a 5% change in methylation over 50 years. So it's a really small effect. And what this means is that since the majority of the CPGs start out unmethylated or methylated, that actually we're really far from the saturation. We never reach these saturation levels. For the great majority, we don't. This is one exception where there seems to be evidence for saturation, but this is also the one that is there. It's the biggest effect size, okay? The great majority are actually in the linear regime, so you can approximate this by a linear process. This is how the methylation, probability of methylation increases. So you can then extend this to the blood tissue. You can generate simulated data, use that simulated data then to train the machine learning models. And this is the end result. So this RR2 index, as you can see, it's higher for the Yang clock, which is more accurate for chronological age, compared to the Horvath clock, which is higher than the pheno age. And so what this is telling us is that the more accurate a clock is for chronological age prediction, the more it could be driven by a stochastic process. Now, if you're surprised by that, you shouldn't, because it's actually just an embodiment of the central limit theorem, okay? So you can have a stochastic process at a single cell level, but what we are doing is we are averaging the methylation over a million cells, a very large number of cells. So that stochastic process becomes deterministic. So it's not surprising that if you had a purely stochastic process, that you can predict chronological age. So that, in a sense, is actually quite trivial. It's really quantifying this, which is non-trivial. Okay, so the converse then is whether the biological age accelerations that we observe, whether this is driven by non-stochastic processes or not. So we took 3 case examples. So we looked at Horvath clock acceleration in males, pheno-age acceleration in COVID-19 cases, pheno-age acceleration in smokers. Yeah? When we replace the clock by the stochastic version, that association disappears, suggesting that these particular instances of biological age acceleration is due to non-stochastic processes. And what is it? Well, the answer is changes in immune cell type composition. So in this paper in 2023, we showed that women have a higher naive T cell fraction than men. This was actually quite a surprise to us because it's not been widely reported in the literature, or we were not looking in the right place. But it's actually a very strong signature. It's as striking or as strong as the association with age. So this is what's driving this biological age acceleration in the case of Horvath's clock. In the case of COVID-19 disease severity, it's even clearer because there you've got an acute inflammation, neutrophil to lymphocyte ratio, which is increased. And that explains why you have this acceleration. Okay, so that clearly, this change in cell type composition is a non-stochastic process. So that makes sense. Now, there's an exception to this, which is that if you look, for instance, at mitotic clocks that estimate the mitotic age in solid tissues, and this is an example here, we've got 6 different tissue types. We're comparing normal breast tissue here to normal adjacent tissue. So this is tissue that is 3 centimeters away from breast cancer. You can see that this clock is always accelerated. Yeah? And if we replace this mitotic clock with a stochastic version, it's also still accelerated. So this is telling us that once you focus on those CPGs that change, the methylation changes with cell division, that it is then mainly, it could be described by a stochastic process, okay? And that makes sense because the tissue at risk is thought to be characterized by an increased mitotic rate, right? So it's an increased rate of that underlying process. Okay, so the last part of the talk, I'd like to tell you a little bit about our adventure in using deep learning methods to try to understand a bit more about the aging process and also about aging clocks. So, as you may know, there have been already attempts to build clocks based on deep learning methods. And what they found was that it didn't really lead to a substantial difference. I mean, it didn't lead to a statistically significant improvement over a simple elastic net regression model. And actually, that's not surprising because they treated the DNA methylation data as a matrix without structure, right? So for deep learning to add value, you need to give the data some structure, be it in a temporal context or be it in a spatial context. And I would also like to highlight this preprint by Wolfgang Huber and Simon Anders because they highlight the point that too many papers are using deep learning methods and not benchmarking them to other machine learning methods. And I think this is really important because, as they showed, it's not really doing better than simple linear methods. Okay, so how do we give the data spatial structure? So we've used PPI networks in the past. So this, because it's a Gerophysics meeting, I thought I mentioned that we've used diffusion entropy to estimate stemness and dedifferentiation at single cell resolution. We've used spin glass algorithms to identify age-associated hotspots, and we've also used transcription factor regulatory networks and mapping TFA changes at single cell resolution. So this is just to advertise this work for people to look at if they're interested. And so we teamed up with Huang De Shuang from in China, We applied a deep graph convolutional neural network with an experimental AI tool. And to derive a clock, a transcriptomic clock, we applied this to over 1 million immune cells from 1,000 donors. Okay, and so this is the end result. This is the highlight. So we found a ribosomal module where the expression of these genes generally goes up with age, and it's independent of cell type. Okay, so you can see that for the four main immune cell types that we looked at, where we had large numbers of cells, this module is changing with age, and it's also changing in brain cell types and in mouse. So it seems to be a universal marker of aging. And this is just a hypothesis about what this may mean. I would be delighted to talk to biologists about this. I don't have time to explain why, but I think that this actually reflects a shallowing of the epigenetic landscape. So this is one of the potential, what it potentially means in terms of biology. Okay, so with that, this is the take-home message, but since this is going to be put on YouTube, I'll just pass this slide. And acknowledgements, well, many people to thank, and my group in particular for all their hard work. So I'll take questions. Many thanks.
Yeah, we have time for questions. Please, go ahead.
Okay. Hi, thank you, Andrew. Really great talk. So I basically have two questions, one about DNA methylation and two mRNA methylation. So the first about the DNA methylation clock. Do you find the methylation singular increase, like the site increase or frequency increase during aging? Or what's the change globally?
You mean globally, or for the distribution of CPG sites? I actually showed you the figure, right? So what I showed you in one of the previous slides was the distribution of the effect size.
They're quite different.
Huh?
They are different between the cell type, right?
No, I don't think so. No, we've looked at monocytes, we've looked at T cells, we've looked at neutrophils. I mean, there are many sorted data sets out there. And the overwhelming majority of the DNA methylation changes are really, really small, right? I mean, we're talking about 5% changes in methylation over 50 years. So it's only a small fraction of the cells that are changing. It's only a small fraction of cells that are changing. And this does not depend on the cell type, no.
Okay, thank you. And the second, what do you think the potential of mRNA methylation clock during aging?
Yeah, that's an interesting question. We've not looked at the mRNA methylation at all. I'm not even sure there is enough data. Has anyone reported changes in mRNA methylation with age? I'm not sure. Probably somebody has, I don't know, but it's not, I doubt that this would be as accurate as a DNA-based clock, right? Anything DNA-based is going to be much more reliable.
Maybe mRNA is more intrinsic, a lot stable. Our previous lab studying the mRNA methylation during the in the blood system, studying the hematopoietic stem cells mRNA methylation pattern, quite similar. We saw the mRNA, the cell type using the bulk, and then use the single cell level to detect the pattern. Thank you.
Right. Okay. Thank you. Mm-hmm.
Okay, thank you. You don't have much time, just one short question. Briefly, what are the main challenges with the clocks, the way you see it? Because it's a very large field now, a lot of controversy, lots of agreement and disagreement. What are the main blocks and what's the main challenges?
Well, one reason I focused on cell type composition, cell type heterogeneity, is because that's certainly one big challenge. I mean, we heard yesterday from Matt, right? He was showing how these estimates, right, these age acceleration estimates varied so much between clocks and also for the same clock within 1 or 2 week intervals, right? This is, in my opinion, driven mainly by the fluctuations in immune cell fractions. They are huge. I mean, if you look at how they change in one person, it's really very, very big fluctuations. But of course, there are changes in the mean, and it's the changes in the mean that are physiologically important. So I think we need to find a way to identify relevant cell types. And I mean, the hope is that the technology is going to advance to the degree where we can do this at single cell level, right? Because that gives you an unbiased way to identify the cell types. And sparseness, as you know, is an issue. So I think that's one thing. And the other thing is we need to probably introduce standards. We need standards because these clocks don't give you an estimate, that doesn't tell you whether the difference or the age acceleration estimate you get, whether it's statistically significantly higher than what you would expect for an average person in the population, right? So if you get this result from True Diagnostics or other companies, they don't tell you. They just say it's higher or lower, but what does that mean? Because you need to have an uncertainty quantification. So there are many issues. I mean, these are just some of the statistical issues, yeah.
Thank you very much.